everyone, welcome back. So this is the second last class for this particular course. Okay, this is the NPTEL course, Experimental Biochemistry. I hope you all are doing very well. So this is the week 11 Experimental Biochemistry class, and in today's class we are going to talk about protein ligand interactions. Okay, so let's start. So we are going to, hello, good evening to you. Yeah. So in today's class, we are going to talk about some of the quantitative methods of protein ligand interaction. Okay, and in the next class, that is tomorrow's class, it will be a revision class for you. Okay, so in this revision class, we are going to go through all the things that we have learned until today, and I will also include some part from the next week's class. Yes, tomorrow it will be an extra class. You will get the mail. Okay, so this extra class will be tomorrow again from 6 o'clock only. So you will get the mail according to that, and it will be a revision class for you guys. Okay, and for the next week's class, it will be our last class for this semester. Okay, and we'll talk about Western blot and other qualitative methods of detection of proteins and protein ligand interactions. Okay, when the ligand is also protein. Okay, so let's start with today's class. Right. So let's talk about protein ligand interactions. Now, protein ligand interactions are one of the most important biological interactions that go on inside our body. We will talk about all the different examples of those interactions as well. Now, protein ligand interactions refer to the binding interactions between a protein molecule and a small molecule known as a ligand. In the biological systems, proteins often interact with various ligands, which can include small molecules, ions, drugs, or other proteins. So these interactions play very important roles in numerous biological processes, including signal transduction, enzymatic reaction, transport of molecules across the cell membranes, regulation of gene expression, etc. Okay. Now the binding of a ligand to a protein typically occurs at very specific sites on that protein. That is called the binding site. Okay. Now this ligand can be a DNA molecule, it can be a sugar molecule, it can be another protein, it can be nucleic acid, it can be anything. Okay, it can be a small molecule. Okay. Now these are called active sites or binding sites, and these binding sites are more or less complementary in shape, size, and chemical properties to the ligand, allowing for very specific recognition and binding. The binding process is often characterized by high affinity and specificity, with the protein binding tightly to its ligand under physiological conditions. Okay, so we will see what are the different interactions that are there between this protein and its ligand. Okay, so even an antigen antibody interaction, it can be considered as a protein ligand interaction where the ligand is also another protein. Right, so there can be different interactions such as hydrogen bonds, ionic bonds, hydrophobic interactions, van der Waals interactions, ionic bonds, etc. Okay, so we will look at this in a little more details. So see, all the different interactions are explained for you. So besides all of these interactions, there can be pi-pi interactions and pi-cation interactions as well. And these are mostly mediated by the aromatic residues. Okay. Now the type of binding. This can be rigid body binding. It can be induced fit where the protein changes conformation upon binding, and it can also be conformational selection. So that means the protein exists in multiple states and the ligand selects only one state, okay, of the protein, and then it binds to it. So some of the natural protein ligand interactions can be, you guys know, it is hemoglobin and oxygen interaction. Okay, then there can be interaction between an enzyme and a substrate. There can be interaction between DNA polymerase and DNTPs. Okay, so that is involved in DNA replication. So any kind of interaction between two or more entities can be considered as a protein ligand interaction if one of them is a protein. Okay. Now there can be this other interactions, for example, ribosome and tRNA interaction, which is important for protein synthesis, right? Transcription factor and DNA interaction, all of these different interactions. Then acetylcholinesterase, which is an enzyme, and acetylcholine, which is a neurotransmitter. So this particular interaction, Right? So all of these different interactions are very important protein ligand interactions. Now, a detailed summary of all different types of protein ligand interactions is given for you. So it can be enzyme substrate interaction, it can be receptor ligand interaction, right? For example, insulin binding to insulin receptor on the cell membrane for the uptake of glucose. So that is a very important receptor ligand interaction. Then transport protein and substrate interaction. For example, hemoglobin and oxygen, myoglobin and oxygen. Right, antigen antibody interactions you already understand. Then there can be ion channel and ligand interaction. So a lot of ion channels are there which are ligand gated ion channels, right? You know about them. So an example given to you for this kind of interaction is acetylcholine binding to nicotinic acetylcholine receptors that allow the passage of ions and initiate muscle contraction. Okay. Now, there are many other examples. For example, transcription factor binding to DNA. There can be enzyme and coenzyme binding. Okay, then there can be microbial signaling, toxin receptor, etc. All of these different interactions are natural interactions where receptor and ligand, so protein and ligand bind. Hormone and receptor interaction, neurotransmitter and receptor interaction, etc. etc. So, drug and natural target molecule interaction. These are all different interactions. Now, how do these interactions happen? So, protein and ligands interact through a process known as molecular recognition. So, molecular recognition involves specific and reversible binding between a protein and a ligand, which can be small molecule, another protein, nucleic acid, lipids, etc. So, this interaction is crucial for various biological processes that you know. Now, binding site. So, if you remember the chapter from enzymes, there we have studied about binding site, right, or the active site. Here also for this protein ligand interaction, there is a specific binding site which is more or less complementary to the structure, shape and size of the ligand. Okay. Then there is complementarity, right? It has to be complementary. Non-covalent forces are mostly at play because it should be reversible. It cannot be irreversible interaction. Then it would become this inhibitor and enzyme interaction. We don't want that because this is a natural system, and in natural system everything is more or less reversible, right? We want to use that particular protein or that particular ligand again later on for some other cause in the body. So all the interactions are through non-covalent interactions, non-covalent forces. Okay. Now, specificity, of course, all the interactions are very, very specific, right? And then binding affinity. So more or less the binding affinity is strong. So binding affinity refers to the strength of the interaction between the protein and the ligand. It is influenced by factors such as complementarity of the binding site and ligand, as well as type and strength of the non-covalent interactions involved. Then induced fit. We talked about induced fit just now, right? So you know all of this. Now, kinetics. So protein ligand interactions are dynamic processes characterized by association and dissociation. So reversibility. The rate of association and dissociation determine the overall affinity of the interaction. So this is something that is very important, and we will read about this in the upcoming slides. Okay, that there is a K on and K off rate. So influencing the biological effects of the ligand on the protein. Okay. Now, there are different techniques that are used for studying protein ligand interactions. Okay. A lot of techniques are listed here for you. So we'll start with X-ray crystallography. This is one of the techniques in which you can visualize both the ligand and the protein and how they interact. Okay. So X-ray crystallography is a powerful qualitative technique to determine the three-dimensional structure of protein ligand complexes at atomic resolution. By crystallizing the complex and subjecting it to X-ray diffraction, researchers can deduce the spatial arrangement of the atoms in the complex, providing greater detailed information about the binding mode and interaction between the protein and the ligand. Okay. Now, another thing is NMR spectroscopy. This is also a structural technique. So, this can be used for both quantitative and qualitative interpretation of a particular protein ligand interaction. So, NMR spectroscopy allows the researchers to investigate the interactions between the protein and ligands in solution. By observing changes in the NMR signals upon ligand binding, researchers can determine binding affinity, stoichiometry, and structural details of the complex. NMR also provides information about the dynamic changes in the protein conformation upon ligand binding. Okay. Now, these are very specialized techniques. Okay. But the everyday techniques that you use, what are they? So for qualitative detection that whether a particular protein is interacting with a ligand or not, for that we can use Western blot if the binding partner is a protein. Okay, so Western blot can be used, pull down assays can be used, chip or DNA footprinting assay if the ligand is a DNA. Chip is chromatin immunoprecipitation, and then we have co-immunoprecipitation and etc. These are all qualitative assays. So whether the binding is happening or not. Now if it is happening, how tightly is the binding happening? So for that we have surface plasmon resonance, we have isothermal calorimetry or ITC, we have fluorescence anisotropy, EMSA, electrophoretic mobility shift assay, etc. So we are going to talk about all of these methods in details today. Okay. Now, let's start with surface plasmon resonance. So these are the techniques from which a lot of questions are asked in GATE, NET, DBT, ICMR, etc. examinations. Okay, so pay attention and go through these topics in details. Okay, so all of these biophysical techniques are very important for any exam that you attend. Okay, so we'll start with surface plasmon resonance. Okay, so SPR is a label-free technique. What does that mean? That means that the protein and the ligand, we do not need any chemical labeling. For example, for EMSA, we need this fluorescent label or radioactive label attached to the DNA. Right, but then for surface plasmon resonance, labeling is not required. Now, why label-free? Being label-free is important because labeling is a very expensive technique. Okay, if it is once getting labeled, even for NMR, you can label the different things that you want to observe, and labeling is very expensive, right? So if it is label free, then you can use it for a lot of different techniques, and it is also relatively cheaper. But SPR is not a cheap technique because this uses gold. Okay, so we look at it right now. So SPR is a label-free technique used to study biomolecular interactions in real time. Okay, in SPR, one binding partner. So what is real time? That means as the reaction is happening, you can observe it, right? Okay. So in SPR, one binding partner, that is typically the protein, is immobilized on the sensor surface. So you can see in this figure that this is the sensor surface and it is coated with gold film. Okay. And on that, the protein is immobilized. So the protein is shown in the form of these tiny Y-shaped molecules that you can see. This is the protein. Okay. It is immobilized on the sensor surface while the binding partner, that is the ligand, this is made to flow at a very slow rate. So this is flown over the surface. Now light comes and it is getting reflected, right? But then when this ligand is binding to the protein, the reflection is different because the refractive index changes. And due to this change in the refractive index, we can actually monitor the binding affinity of this particular protein for the ligand. Okay, so the changes in the refractive index at the sensor surface due to the complex formation is monitored, allowing for the determination of binding kinetics, affinity and specificity. Okay, so surface plasmon resonance helps perform molecular interactions in real time. The studies can be used to observe if two or more interactants bind to each other. It is used to measure how strong the interactions are and even measure the actual association and dissociation rates. So K off and K on, these can be measured. Specificity can be measured. Affinity can be measured from this interaction. So to perform, this is used for performing protein-protein interaction and then to analyze the molecular interaction obtained as an SPR curve or a sensogram. So the figure that you see on the right, this is called a sensogram. Okay, the figure that you see on the right, one second, that is called a sensogram. So to calculate the binding affinity of the protein interactions performed. Okay, so So now if we look at the sensogram, we will see that initially there is baseline and then you load. So what happens? You get some value, some response. Then again, this is only because of the protein. Then you load the ligand. And what happens? The refractive index changes, and that is why the response is different. And that is why you get even higher peak, and that is called the association phase. Next, what happens is all the protein molecules are saturated with the ligands. Then even when you're flowing more ligands across it, it is not binding anymore, and that is giving you this saturation. Then there is the dissociation phase, and then once again you have to regenerate and the baseline is reached. Okay, so buffer, sample, buffer, analyte, then buffer, and then regeneration buffer, again buffer, right? So that is how it goes. Now we will look at this in more details. Okay, so one interesting thing is that what is this plasmon and why is gold used, right? So these are the questions that would definitely come to your mind. So interestingly, plasmon is the free electrons on the gold. Okay, so you know that gold is present at very low position in the periodic table. So that means it has a lot of electrons in it, right? And all the free electrons, the valence electrons, these are very mobile. So gold is a very good conductor of electricity. This we know. That means that when electricity is flown, then all these free electrons, they are actually moving and they are carrying electricity, right? So these electrons, they are only called plasmon. So let us look at what happens exactly and why exactly do we need gold. Right, so surface plasmon resonance is a phenomena that can be observed in thin conducting films at an interface between media of different refractive index. So in the SPR system, the media are glass of the sensor chip and the sample solution. And the conducting film is a thin layer of gold on the sensor chip surface. So SPR is a physical process that can occur when plane polarized light. What is plane polarized light? The light in which the electric and magnetic field, they are vibrating, and they are vibrating perpendicular to one another but along the single plane. Okay, so all the electric field vectors, they are vibrating along the single plane. So that is plane polarized light. It's a thin metal plate under total internal reflection condition. Now you know what total internal reflection is. For example, this is the surface. This is the angle at which it hits. It is more than the critical angle, and because of that all the light is reflected back, right? So there is no light that is lost or refracted. So all the light is reflected back, right? So total internal reflection is happening. When a light beam hits a half circular prism, the light is bent towards the plane of the interface when it is passing from a denser medium to rarer medium. That is the characteristic of total internal reflection. So this is the rarer medium and this is the denser medium, right? Now let's see what happens. So changing the angle of incidence changes the outcoming light until it reaches a critical angle, which is your IC. At this point, all the incoming light reflects within the circular prism. This is called total internal reflection, right? Now, the surface is coated with a thin film of noble metal. Okay, not reactive. Noble meaning not reactive. Usually gold. So gold is used in the sensor chips because it combines favorable SPR characteristic with stability. So gold is not reactive. So that is one of the very important characteristics. With stability and a high level of inertness, it does not interact with the proteins also that immobilize on it. Right. In the biomolecular interaction context, at a certain combination of angle of incidence and wavelength or energy, the incident light excites the plasmons. So what are these plasmons? Electron charge density waves in the gold film. As a result, a characteristic absorption of energy via the evanescent wave field occurs and SPR is seen as a drop in intensity. So the intensity of light is dropped of the reflected light as shown in the figure. Right. So you can see here in an in SPR experiment, one molecule, that is a ligand, is immobilized on the sensor chip and binding to the second molecule, which is the analyte, is measured under flow. So response is measured in resonance units or RU and is proportional to the mass on the surface for any given interactant, and the response is proportional to the number of molecules bound to the surface. So that is how you know the stoichiometry also. So response is recorded and displayed in a sensogram in real time, the one that we saw. Okay, so how is this ligand immobilization done? Covalent immobilization using amine coupling method. So that is explained for you. Okay, right. So that is it about SPR. Okay. Now we will look at another method which is relatively very simple. This is chromatin immunoprecipitation. So here what happens? So that one was between a protein and another protein, or a protein and a ligand. Let's say small molecule, right? SPR. You can also do it between a protein and DNA, but then a lot of the thing would be required. Okay. Now, chromatin immunoprecipitation. So what happens here? Let's say this is a protein. It is bound to DNA. Now we want it to be bound to DNA and not get separated from DNA. So what we do is we cross-link it. Covalently cross-link this. So whatever happens, this is not going to get removed from this DNA. And all the regions of the DNA that are not bound to the protein, those are then removed. Okay, and only the portion that is bound to DNA, that is examined. So you can remove it if it's a technique called DNase footprinting. Then you can use DNase to chop off all the free DNA. Okay, and you can take the protein and like from it you can reverse this cross-linking and the protein will be free then, and then you can study, okay, so this is the protein that was bound to my DNA, right? But in chip what happens, let's see. So chromatin immunoprecipitation or chip is used to study protein DNA interactions in vivo. So in vivo means within the organism, right? Within the body of the organism. So in vivo, and proteins are, so it can be even within bacteria. Okay. So proteins are cross-linked to DNA, fragmented. So the DNA is chopped off and selectively enriched using specific antibodies. So the antibody specific to that protein that comes and binds to that protein. The associated DNA is then identified and analyzed. So that means you can understand the binding site of a specific transcription factor, binding site of a specific enzyme if that is a DNA binding enzyme. Okay, so all of these things, for example, T7 RNA polymerase, it binds to the promoter DNA that we know, right? Let's say something binds to the enhancer. So all of those different proteins, if you have antibodies against them, you can know the binding site. So the thing is explained in this diagram. So inside the cell, you cross-link it covalently and then antibody binding is done. Antibody binds to the protein of interest, it is washed, and then cross-link is reversed, so the protein remains bound, the DNA comes, and then you sequence this DNA. You do DNA purification, quantitative PCR, and then you do protein and DNA analysis. You will get to know the DNA sequence. Okay, so the procedure is cross-linking. The cells are treated with formaldehyde and covalently cross-linked. Protein DNA complexes are formed. Chromatin fragmentation. So DNA is sheared by sonication and enzymatic digestion. Then immunoprecipitation, that is antibody specific to the target protein is used. Protein DNA complexes are pulled down. And reverse cross-linking. So protein DNA cross-links are reversed and DNA purified. Finally, DNA analysis is done using PCR and quantitative PCR. Specific regions are amplified, and then sequencing is done. For example, chip sequencing is something that is a genome-wide binding profile. For example, for a particular protein, it tells you exactly where in the genome it binds. Okay, everywhere in the genome, wherever it binds. Okay, so there are three different variants, that is chip PCR, chip qPCR, and chip sequencing. So that is all about chromatin immunoprecipitation. Now we will talk about another very important technique that is called isothermal titration calorimetry. So what is happening here? Okay, let's say there is this protein. You add the ligand. Because the protein is binding to the ligand, there is some amount of, since the bonds are forming, so it is either a exothermic reaction or endothermic reaction, right? So some amount of heat energy is either released or it is absorbed, and that change in heat energy is actually detected, and from there you calculate exactly what is the stoichiometry, what is the binding affinity, and everything else, okay, of that particular protein for that particular ligand. So how are we doing it? Let's see. So ITC is a technique used to measure the heat released or absorbed during a bimolecular interaction by titrating a solution of protein with a ligand and measuring the resulting heat changes. Researchers can determine thermodynamic parameters such as binding affinity, stoichiometry, and enthalpy of binding. Okay. Now, isothermal titration calorimetry is one of the very important techniques used to determine label-free specific interactions between molecules dissolved in aqueous solutions such as protein ligand binding, protein-protein binding, DNA-protein binding, protein-carbohydrate binding, protein-lipid binding, antigen-antibody interaction, etc. This is a very sensitive technique that works by titrating a binding reagent that is known as the titrant, generally a small chemical compound called the ligand, into the titrant, usually protein at a constant temperature. So this technique is commonly used for drug development as it provides valuable thermodynamic information about the interaction of the molecule with the target protein. So ITC can precisely determine the Gibbs energy, Gibbs free energy, enthalpy, entropy, heat capacity changes associated with the binding or interaction with the biomolecule. So let's look at the setup. So there is a reference cell and there is the sample cell. So you provide some amount of energy so that the temperature is same in both the reference cell and the sample cell. Now you add the titrant. There is some temperature fluctuation. So what you do is, let's say you have to provide more heat into the system, and now once again the reference cell and sample cell are having same temperature. Then you add the ligand. Once you add the ligand, once again there is some temperature difference. Let's say this temperature is reduced. So that means it is an exothermic reaction, right? And then what is happening? No, let's say the temperature is increasing. So that means it is a what? Endothermic reaction. Then what you have to do is you have to change the temperature of the reference cell once again. And this difference in temperature is actually monitored, and this difference in temperature is used to ultimately calculate the binding affinity and everything else. So we will look at this in details now. So this is called the, this is the raw data that you're getting, and this is the final binding curve that you're getting. Okay. So we will interpret this as well. And you have questions on that. Okay. So this ITC instrument measures the amount of power in microcals per second required to maintain a constant temperature difference close to zero between the sample and the reference cell. Initially, the feedback system continuously applies a small power to the sample cell, which determines the baseline level. Each injection of the syringe, usually termed as the ligand, triggers the binding reaction depending upon the binding affinity and concentration of the reactants, that is macromolecule M and ligand L in the cell, and certain amount of macromolecule ligand complex or ML. The formation of the complex is accompanied by release or exothermic reaction or absorption, that is endothermic reaction of the heat that causes a difference in the temperature between these two cells, right? Now what happens is, then the feedback system either lowers or raises the thermal power applied to compensate for such temperature imbalance. Then what happens is, so then after each injection, the system reaches equilibrium and the temperature balance is restored. So once again equilibrium is reached and the temperature balance is restored, right? So now, so the temperature balance is restored. Okay. So therefore the recorded signal shows typical deflection pattern in the form of a peak. Okay, so all of these tiny lines that you're seeing, these are all peaks. Okay, so integrating the area under the peak, assuming the baseline as a reference provides the amount of heat associated with injection. As the reactant in the cell becomes saturated, the heat signal diminishes only until the background heat. In a properly designed typical ITC experiment, the binding affinity or dissociation constant Kd or binding constant Kb, that is 1 by Kd, stoichiometry N, enthalpy delta H at a constant temperature T are measured. From this data, your delta G, that is the Gibbs free energy, change in the Gibbs free energy, and then entropy change may be calculated if one protein molecule M binds to one ligand, then 11 binding modulus, then the binding module is applied. 1:1 binding module is applied. Okay. Now, we will see how to interpret this particular graph. Okay. Now see. So the raw data is called the thermogram. Top panel. Okay. This whatever you see. So the plot of the heat flow, that is in powers and microcals per second versus time, that is shown. So each peak is equal to 1 ligand injection. So exothermic interaction, since the peaks are all downwards, it is exothermic reaction. If they would be upward, it would be an endothermic reaction. Okay, so that means heat is released when this ligand binds to the protein. Okay, if it would be upward, then it would be endothermic reaction. Then we have saturation. So the peak size decreases progressively, that is saturation. So eventually returns to baseline, that is no further binding, right? What we saw in the sensogram of the SPR as well. Now, integrated plot. This is called binding isotherm. That is called thermogram. This is called binding isotherm. So what happens here? Plot of heat per injection, that is delta Q versus molar ratio, that is ligand by macromolecular protein, right? So ligand by protein. So the plot of this two will tell you what, right? So sigmoidal curve, if it's a sigmoidal or S-shaped curve, that tells you that the binding increases progressively and ultimately undergoes saturation. The midpoint tells you about the stoichiometry. So that means if it is 1:1, then that means the interaction is once again stoichiometry is 1. Okay, and then the slope of the curve, this reflects the binding affinity. Height of the curve, so steeper slope means higher affinity. Height of the curve tells you about the delta H, that is total vertical change is equal to enthalpy of binding. Okay. Now hopefully you will be able to see all the arrows are shown here for your understanding. This Ka is equal to 1 by Kd, which is the association and dissociation constants, and this is the stoichiometry. Okay, so midpoint is stoichiometry. Now we have a question. Let's try to answer this. Okay, there are 3 questions. If delta H is negative and delta S is also negative, binding is favored when temperature increases. So you have to remember the equation, thermodynamic equation, that is delta G is equal to delta H minus T delta S. In that equation, when is binding favored? Okay, what happens to temperature if binding is favored? So binding is favored means delta G is negative, correct? So from that you can answer B. Okay, we look at the other 2 as well. So which combination gives the lowest Kd, that is the strongest binding? High Ka, high Kd. Low Ka, low Kd. High Ka, low Kd, low Ka and high Kd. C, high Ka and low Kd. Good. Yes. So now chip sequencing is most useful for B. Great. Let's see. Let's look at the right answers. Yes. So you can see that when temperature decreases, your delta G becomes most favorable. Correct? Because what? Delta G is negative and delta S is also negative and temperature decreases. So then this term becomes more, and that is why delta G will again decrease more and it will become more negative. Okay, so the other two are also correct. Now we will move on to another technique, which is fluorescence anisotropy. Okay, so fluorescence anisotropy or fluorescence spectroscopy can be employed to study protein-ligand interaction using fluorescently labeled proteins. Now here proteins are labeled. Either protein is labeled or maybe your DNA is labeled. Okay, at least one of them have to be labeled. Okay, by monitoring changes in the fluorescence intensity, polarization or lifetime upon ligand binding, researchers can determine binding affinity, kinetics, conformational changes in the protein. So what is happening? Let's say initially there is depolarized light. It is being passed through the polarizer and then there is polarized light. Now when this polarized light comes and hits a protein DNA complex, what happens? Since this is a large molecule, there is very slow tumbling and polarized light is only obtained. But when only the ligand is there, ligand is moving very fast, it is fast tumbling. What happens? More depolarized light is obtained. Okay. Now the principles of using fluorescence anisotropy is to measure protein-ligand interactions. Free fluorescent ligands or peptides irradiated with plain polarized light rotate light very quickly and emit light in a depolarized manner. Initially all the light was here, later on all the light can be anywhere, right? The electric vector can be anywhere. Now fluorescent ligands bound to larger proteins, they tumble very slowly and emit more polarized light. The degree of molecular binding can be inferred by measuring the amount of polarized emissions. Okay, now how do we interpret the plot? So the raw data, that is polarized fluorescent signal, is measured as parallel intensity, that is I parallel, and perpendicular intensity, that is I perpendicular. Anisotropy is calculated as I parallel minus I perpendicular, intensity of light this is, okay, and divided by I parallel plus 2 into I perpendicular. So this I parallel is fluorescence intensity parallel to the plane polarized excitation light, and I perpendicular is the fluorescence intensity perpendicular to the excitation plane. Okay. Now the binding curve, that is the main plot, the plot of the anisotropy versus protein concentration or ligand concentration. This is once again your S-shaped plot. Okay. So the total intensity is equal to I parallel plus 2I perpendicular, because emission occurs in 3-dimensional space. So 2 perpendicular component exists relative to the excitation polarization. Okay, so you can read about this if you're having any doubts, and then we can talk again if you have any doubts in the next class about this. Okay, so now we have EMSA. So in EMSA, what is happening now? EMSA is once again very similar to fluorescence anisotropy, because here also the final plot is after all a KD plot. Okay, this S-shaped KD plot. So here what is happening? Let's see. There is this protein. Protein binds to nucleic acid. Nucleic acid is labeled. It can be 5' fam labeled, or any other chemical group can be there that is fluorescently labeled or radioactively labeled. If it is radioactively labeled, it is more sensitive. If it is fluorescently labeled, there is less chance of you dying. Okay, so now people are going more and more for fluorescent labeling, although it is less sensitive, but this is safer for the researcher. Okay, so now Kaon is the units of Kaon. Kaon is the rate at which the protein and RNA forms a complex. What is K of? The rate at which they dissociate. Okay, so Kd, remember, is equal to K of by Kaon. Rate of dissociation is equal to K of by Kaon. That is Kd. Then Ka would be Kaon by K of. Correct? Now, what do we know? So this is how the gel looks like. Okay. there is free mRNA, and then if you look at this particular lane, this one, you will see that there is this tiny shift, right? So why is that shift coming? That shift is coming because the protein is bound to RNA. It is getting retarded because protein is heavier than RNA. So RNA, normal RNA, it runs faster. But then if you keep on increasing the protein concentration, more and more protein molecules will come and bind to the RNA. It will retard the mobility of the RNA. That is why you see two different bands. Now, at the point where the intensity of the two bands is equal and the intensity of the upper band is equal to the half of the intensity of the free RNA band, then it is called, not the upper band, the lower band or upper band, any one, it is half the intensity of the free RNA band, that is called Kd. So what is Kd? Kd is the concentration of the protein at which half of the protein molecules are bound to the RNA. Okay, so half of the binding sites are occupied. That is called Kd. So in this entire story, what all do you know? Okay, some of the components values you know, and for some of the components you do not know the values. For example, P total you know, because how much protein you are adding into every lane, that you know. How much RNA you are adding into every lane, that also you know. But how much RNA is binding to how much protein, that we do not know. Correct? Kd we do not know as of now. Right. So how much is forming complex, that we don't know. Correct. So you know, and how much is free, that also we don't know. So we only know P total and R total. So now fraction bound. So amount of the total protein that is there, that is in the denominator, and amount that is bound, that is in the numerator. Right. So we don't know fraction bound also. Now from there we derive that Kd is equal to R free into protein free divided by protein into R. We do not know anything about all these values, which is again from here also you can know that fraction bound is equal to P total divided by P total plus Kd. Okay, so P total we know. Fraction bound we can derive, and Kd we can calculate or we can estimate from this image. So there is this image analysis software which is called ImageJ, and using this software so from here, from this gel we can say that yeah, Kd is 3.5 micromoles of protein, right? But that is something that I am just looking at it and saying. But then there is a full software called Solver, which is there in Excel, MS Excel, and using this particular software, when you put the values and when you get the full graph, from there you can actually calculate the assumed Kd, calculated Kd, and then from there you can get the actual Kd value for that particular gel. Now you will have to run at least 10 gels or 20 gels and then get the best Kd values from there, that are best gels and most reliable Kd values. Take an average of them and finally report that this is the binding affinity of that protein with a particular DNA or RNA. So this is something that we do in the lab regularly. Okay. Now for a binding reaction, let's try to solve this question. For a binding reaction, protein P plus ligand L, that forms PL complex, the unit of K on and K off have the units. Okay. Dash and dash respectively. You can answer in the chat box. D. Okay. Good. Now this is an important question. You can expect this in your exams as well. Okay. So it is mole inverse, second inverse, and second inverse. Correct. Now we have another question. Which of the following experiments can be used to determine the binding kinetics under favorable conditions? So Electrophoretic Mobility Shift Assay, Isothermal Titration Calorimetry, Fluorescence Anisotropy, and surface plasmon resonance, which would give you the binding kinetics under favorable conditions. Binding kinetics means Kd. Okay, let's look at the right answer. So all the given answers are correct. So we saw that all of these are quantitative techniques, right? So that means they will tell you the binding kinetics. So that means the Kd value. What is this binding kinetics? This is the affinity of the binding, that is K on K of all of this, and the ratio of K off by K on is Kd. Correct. So which of these techniques will tell you how tightly the ligand is binding to the protein? So all of these will tell you, because they are all quantitative techniques. Right. Now we have another question. Enthalpy of binding reaction can be determined from. This is a relatively direct question. Try answering. Isothermal titration calorimetry, fluorescence anisotropy, surface plasmon resonance, electrophoretic mobility shift assay. A, exactly, right. So enthalpy of binding, so that means anything regarding the temperature, okay, that is given by isothermal titration calorimetry. There is one more thing called differential scanning calorimetry. Read about that as well. That is also very interesting. Okay, so you can read about that. It is called DSC. Now, dissociation rate constant K of for a protein, the DNA interaction is determined by EMSA. The intensity of the free ligand can be fit to which of the following equations to obtain K of. Okay, so these questions, it's best if you practice, because these are gate level questions. And only when you practice, you will be able to answer them. The other question is, which of the following statement about protein ligand interaction is true? Ka, that is the association constant, is independent of conditions such as salt concentration and pH. When all the binding sites are occupied, Ka, that is the association constant, equals to ligand concentration. If Ka or the association constant is increased, then binding will be tighter. The larger Ka means weaker affinity. So you can see that two are controversial, right? So these two questions, they are opposing one. The two options are opposing one another. Okay, so we'll try to look at the right answer. Yeah, so the right answer is A for the first one. So what exactly is this? I is intensity. I naught is the intensity of the light that is initially, so basically I is the intensity of the, you go to a machine, and from there it's called chemidoc, and the fluorescence intensity is detected, right? That is how you get the bands, and the intensity, fluorescence intensity of this band, it is converted into this I value using a imager. It is called image J. So this is a software. This converts it into this image value. So I is equal to I naught into 1 minus e to the power minus k of into t. Okay, so you can read about this more. So what is this happening? What is exactly happening here? Initial intensity that was there when no protein was bound, that is getting, so that is getting reduced by this protein that is bound to it, and that is why it is minus to the power k of into t. Acha. And the next one is Ka is increased, then the binding will be tighter. That is true, but the others are false, because of course association will be affected by salt and salt concentration and pH. Okay, and when all the binding sites are occupied, then Ka is equal to ligand concentration. That is not correct. That is Total is saturation. Okay. Now this is another important question. This is the raw data plot. Okay. Raw data plot of a particular thermogram. Now you can see the total heat can be expressed as. So there are these different Qs, and whether the total heat is multiplication or addition or what is happening. Okay. So we look at the right answer. So it is basically addition of all of these. So the total heat is actually the integration, right? So integration of the area under the curve. So if we go back to this particular slide, we will know. See, I'll tell you, no problem. Yeah. So we come to this slide, right? Now if you look at this, you will see that each peak is one ligand injection, and so the one before this. So if you integrate all the area under the peaks, then you will get the total heat capacity. Okay. So these topics, they are complicated, right? So it is best that you read all of these, read about all of this from a very good review paper or good books. For example, you have Voigt and Voigt, you have Denninger. These good books are there, biochemistry books. Read, but I'm not sure if all of these topics would be there in books, because when I was doing my masters, I had read from review papers, which are very self-explanatory, right? So search these individual topics, ITC, SPR, etc. You'll find a lot of material on them. Read exactly what is what, how do you calculate, how do you interpret these graphs, because these are very important questions. And if you know the basic principle, that is good enough, but you will have to know how to interpret the results also. Okay, so you have to basically get the what? Summation. Okay, it's an integration basically. Right, now we have another question. So this is all about the principles. Okay, hopefully you'll be able to answer this. This shouldn't be very challenging. Let's see. So ITC, fluorescence anisotropy, surface plasmon resonance, and EMSA. Now try to match whatever is in the right hand side column. Okay. So one should be which one? Okay, let's see. Let's see the right answer. Yes. So D is the right answer. Why? Because ITC will tell you everything about thermodynamic parameters. Good. The next one is fluorescence anisotropy. It should be 4. Right? So axis of polarization, that is not there in any other technique. Good. Now surface plasmon resonance. So surface plasmon resonance, the resonant oscillation of conducting electrons at the interface between negative and positive permittivity material in a particle stimulated by incident light. So definitely surface plasmon resonance. And the last one is EMSA. So EMSA is protein DNA complex detection. Right? So this one was doable. Okay, good. Now cytochrome P450 enzyme binds to a newly synthesized drug molecule with an association constant of Ka is equal to 10 to the power 7 mole inverse. What is most likely measured dissociation constant in the presence of a competing inhibitor? So that means you know that association constant is 1 by dissociation constant. But if there is a competing inhibitor present, then what is going to happen to the value? Is it going to increase, decrease? What is going to happen? This is doable. Try. This is not very difficult. So if Kd is 10 to the power 7 mole inverse, then what is Ka? Sorry, what is Kd? If Ka is this, then what would Kd be? 1 by 10 to the power 7. So that becomes 10 to the power minus 7. What is less than that in this question? C. So now A, no, 10 to the power minus 7 is lower or 10 to the power minus 6 is lower? Which is lower? 10 to the power minus 6 is more than 10 to the power minus 7, right? 10 to the power minus 8 is even lower. Okay, so you can look this up whether the answer should be 10 to the power minus 8 or 10 to the power minus 7. You can look this up. Okay, so what should be the right answer? You can try to find out. Okay, so in the presence of a competing inhibitor. Oh, acha, you also have to take into account if Km is getting changed. So whether Km changes or not, that also you have to take into account. So you can actually look this up. Look up and check whether this answer is correct or not. Okay. Achha. Let's go to the next one. If the free energy, that is delta G, for the binding interaction is known, then we calculate what? Which of the options is correct? Association constant, dissociation constant, binding on, off. What can you calculate from free energy? If you remember two equations, you know that one of the equation is delta G is equal to minus RT ln KD, right? So from there you can know KD. Then if you know KD, you will know Ka. So if you know Ka, can you know this binding K on rate and K off rate? Okay, let's look at the right answer. So you can know 1 and 2 because K on and K off, you will know the ratios. K off by K on you will know and K on by K off you will know. But you will not know the exact K on and exact K off from KD. Correct? Achha. Now K on and K off values for the ligands X and Y with the enzyme E is given below. Ligand X and Y, K on value, K off value is given. Okay? So which of the following statements is true? X binds E more tightly, Y binds E more tightly, XE complex is longer lived than YE complex, YE complex is longer lived than XE complex. So this is one of the questions that was there in my PhD entrance exam. That much I remember. So now you will understand how important these questions are for you guys. Okay. Right. So try to answer this. Now which one binds more tightly? From where do you get to know about that? The ratio of K off by K on. That tells you about the KD. If the KD is low, that means it is going to bind more tightly. Correct? And which one lives longer? So if the K off rate, K off is very small, K on is larger. So that means K on by K off ratio will tell you which one remains bound for long. Okay, good. So let's see which is the right answer. Acha, so C is the right answer. Why is C the right answer? Do the calculation, okay, and then we can check. Okay, so K off by K on will give you which complex is binding more tightly, and K on by K off will tell you which one is longer lived. this and then we can check if the answer is wrong or not. Okay. Now we go to the next one. In ITC, a titration experiment for a titration experiment to determine the dissociation constant, that is Kd, it is necessary to vary the temperature. ITC experiment. The ligand concentration is varied and the protein concentration is kept constant. Protein concentration is varied and ligand concentration is kept constant. Concentration of free ligand, free protein bound complex must be determined. So which do you think is the right answer? B. So B is 2 and 3 are correct. Let's see. Yes. So B is the right answer. 2 and 3 are correct. Okay. Okay. Now the researcher designed a new drug molecule and wanted to study the interaction for this drug molecule with human peptidase enzyme. If the peptidase drug molecule complex dissociates with a K-off rate of 1549 per second, then what is the time constant for this reaction? So what are you going to use? If you remember the decay curve, first order decay curve, okay, let's see. Yeah. So tau is equal to 1 by K of. So that is giving you. So how much time is needed? So in 1 second, in 1 second, this much amount is dissociating. So then what is the time constant for the reaction? Right. So then how much time will it take for all of it to decay? So that is tau is equal to 1 by K of, and that is giving you 645.5 microsecond. That much time it will take to completely for the whole of it to decay. Okay. Now dissociation constant K of for a protein DNA interaction is determined by EMSA. The intensity of the free ligand determined corresponding at a particular time interval is given in the table. Determine the K of. How will you determine the K of? You remember that formula that we saw. I is equal to I naught into 1 minus e to the power minus K of into T. Correct? So T is time and I is given. So from there you can calculate the K of L. I naught is not given, but I value is given. So in every minute, how much intensity is decreasing? That way we can calculate. K of is basically 0.1 per minute. Okay. Right. So now we have another one. A very similar. How do you find this kind of things interesting? Oh, this is interesting because the principle is very interesting. Okay, the questions I can understand, they are a little tough, but once you start practicing, they will not look that tough to you anymore. And I will hope that they will not give very tough questions in the exams. And of course, questions will be mixed. So you can attempt to choose some questions and not attempt other questions. Okay, so that is always an option. So we don't need to worry. If some question looks very difficult, we will simply not attempt that, right? Okay. A very similar protein ligand titration experiment was repeated. where the protein concentration of 10 micromolar is used, which is more than expected Kd. The concentration of the ligand and the protein ligand complex for each titration is given below. Determine the dissociation constant. So ligand concentration, protein ligand concentration, that is given. Right? So if you remember the formula for Kd, then you will be able to answer this. Free ligand concentration is given, and you will have to find out Kd and the bound fraction that you will have to find, and then you will be able to answer. Okay. So basically the right answer would be 1 micromolar. Okay, so in that case, in tomorrow's class, we will go through all these questions with proper explanation. Okay, because it is anyway a revision class, and if we will have other questions as well. But since these questions I can also understand, they are a little difficult, so we will try to go through these questions once again. Okay, I think there are like 5 or 6 questions that are tough in today's class, so we will go through each and every question properly with proper explanation. Okay, hopefully that will help you. Okay, so that was it for today's class. Thank you so much for paying attention to today's class. And once again, the links for SPR and ITC are given. Go through them. They are these virtual lab links. Okay, and we will go through all the difficult questions of today's class in tomorrow's class with proper how to solve them and explanations. Okay. Thank you so much. If there are any doubts, anything, then you can mail me. I will give my mail ID once again, and you can mail me if there is anything. Yeah, sure. Thank you. So this is my mail ID. If anybody wants me to solve any questions for you guys, help you with any questions or any concept or anything else, you can always mail me.